So this one's not so bad. Even though it kind of builds up to 4 different lectures, it is kind of really, really intense on how much information we cover in here. The brain introductory, this particular micro lecture in micro lecture 1, will contain the majority of the information you'll see on your test. Because the introductory part is pretty much the main things you need to know. We're going to go through all the different little parts. What are the landmarks? What's the cerebellum, cerebrum? We're going to talk about the most difficult part of this lecture, which is the embryonic development. I'll kind of tell you why in week 4 you might have spinal bifida, because of how it forms that final tube. So we're going to look at that. But the most difficult part about embryonic development is going to be the mesencephalon, metencephalon, and the myelencephalon. What do they all turn into? You're going to have a structure called the diencephalon. What are the structures of the diencephalon? And the only way to do this, I can't stress this enough, make a note card, because these words are so confusing and so close that you're going to get confused, you're going to make silly mistakes. But if you look at this brain slice right here, and if you look at it, do you happen to see anything weird or strange?
Yes.
Yep. You see this kind of structure over here on the side? You see how this tissue looks a lot different than all these other types of tissues? That's just an example of a brain tumor. You don't need to be a doctor to know that shouldn't be there, because in the brain, the left and the right side should be very similar to both sides. It could, if it was a stroke, this could actually be a stroke where you had blood vessels kind of coming in. They either burst with an aneurysm, and then all this tissue is dead, or this could be growth of tissues. And the reason why I say it could actually be a tumor is because you see more growth over here, and it's spreading out over here past this little, there's a ventricle right there that's being pushed down. So if this wasn't a tumor, it wouldn't have been growing into the ventricle. But you still see cell death and stuff in a stroke. So the brain is complex. You don't need me to tell you that. Brain function is associated with life. Your personality, everything that makes you you, is all happening in the brain. And this chapter is going to look at the brain, and then the last micro lecture is going to be cranial nerves, and that's kind of easy. We're going to kind of look at the brain circuitry and the function, but very, very, very briefly. Aristotle, I mentioned it before, thought the brain was a radiator and it cooled blood. He was wrong. Hippocrates was more accurate. From the brain only arises our pleasures, joys, laughters, jest, as well as our sorrows, pains, griefs, and tears. He was a poet at heart, even though he was the father of medicine. But he kind of got it a little bit better, where they say most of our thoughts, most of our memories, most of what makes us us is in the brain. If you don't have any brain activity, or the ending of brain activity, that's the clinical definition of death. So even though you're respiratory, your cells are still, growing and everything, if you don't have brain activity, you are clinically dead. And there are examples of people who are declared clinically dead that come back to life, but they're extremely rare. And usually in every one of those cases, there's something that they missed before they said that they're clinically dead. So when you get in the hospital, you'll see this in the hospital. They do a little water test in the ear, and then what happens is it stimulates the tympanic membrane. That stimulates neural activity, and if nothing is kind of showing up on the brain, that's one of the ways that they can test if they're dead. They also shine light in the eye to see if there's any retinal response. So evolution of the central nervous system shows spinal cord has changed very little. So how we get the impulses to and from our organs and glands to our muscles hasn't changed much, but our brain has. Vision, memory, motor control, and the ability to use our prehensile hand. So we're going to look at the major subdivisions and anatomical landmarks of the brain, describe the location of its gray and white matter, and unfortunately describe the embryonic development of the central nervous system and relate this to the adult brain anatomy. And again, if you understand embryonic development, you can better understand the progression of some of these diseases that affect the brain. So what's rostral? Rostral means to the nose or towards the nose, but in this case, your nose just happens to be towards the forehead, so it's in the front. Caudal means tail, so towards the spinal cord. If you played Mortal Kombat and you defeated that enemy and you ripped the spinal cord out of it, you would see the spinal cord just dangling there like a tail. So caudal, tail. Then you have 3 different parts. You have the cerebrum. Cerebrum is the large part, 83%, all of that, cerebrum. This little thing in the back that kind of looks like a bird's nest of just intertwined fibers, that's the cerebellum. And the brainstem is everything you see in yellow. All of that is the brainstem. So if you kind of think about it, the brainstem is the portion of the brain that remains if you remove the cerebrum and the cerebellum. The diencephalon, midbrain pons, and the medulla oblongata make up the brainstem. One generic way to think about it. I told you the spinal cord didn't really change that much in our evolutionary history. How we get signals from point A to point B didn't really change much. So, if you were making a brain, do you think the midbrain and the pons and the medulla oblongata are probably more for some of the older types of functions we need? And moving kind of up to the cerebellum, those are probably for other types of controls that we don't really want to think about. But when we go all the way further away from the spinal cord, this part of the cerebrum is called the neocortex. It's called neocortex because it's new. This is where most of our personalities, memories, feelings, all of those types of complex things are happening in the cerebrum. So the further you get away from the spinal cord, if you're taking a test and you go blank on everything, the more things that make you you happen in the cerebrum. The things you don't want to think about, like thirst. Do you ever kind of sit there and say, you know what? I feel like I want to be thirsty. No, you just get thirsty and drink water. So these are things that are just going to happen in the background that you're going to take care of, and all of that stuff is going to be happening here. Don't worry, we're going to memorize all that, but if you blank out on the test, there are ways to kind of get around it. Longitudinal fissure for another landmark. Longitudinal fissure runs down the length, and it separates the left and the right hemispheres. You have the gyri, which are the bumps. Sulci, which is the groove. The gyri right here is the primary motor. Primary sensory is back here. We'll go over that later. But some of these gyres have been discovered and have been known for exactly what they do. Other ones, unfortunately, they don't really know. And if I'm being honest, the reason why they know why some of these do certain things is either gunshot wounds, people get stabbed in the head in very gruesome things where they lose function. The primary motor and primary sensory, they actually stuck probes in there and they stopped function from happening. So that's how they kind of know this. And the corpus callosum is a white section of the brain. White is now means myelin, so it's going to connect the left and the right hemispheres, and we'll look at that when we look at the sagittal section of the brain. Brainstem, you kind of saw that. The brainstem is going to have the diencephalon. Diencephalon is going to be all up here. Midbrain, midbrain is up there. Pons is going to be right there. Medulla oblongata is going to be down there. We're going to look in more detail what's happening, or what are the parts of the diencephalon, what's happening in the midbrain, the pons, and the medulla. This is your corpus callosum I kind of mentioned before. We're going to talk about the optic chiasm when we look at the vision, and this is the optic cross where the left signals cross over to the right and the right signals cross over to the left. Here's your midbrain, pons, and your medulla oblongata. There's this little dot right there that's a commissure where they connect the left and right sides, all in this area. It's being bathed in cerebral spinal fluid. That's going to be very important later. And there's your cerebral aqueduct that goes to the fourth ventricle. Also for cerebral spinal fluid, you're going to have the thalamus and the hypothalamus. They're going to be important when we talk about the diencephalon, and then all that good stuff. Depending on what your lecture teacher makes you memorize, you might have to memorize everything or just a couple things. And if you look at the brain, you can kind of see some kind of interesting structures. Like you see these white lines in the cerebellum. Those white lines are called arbor vitae. They're called arbor vitae because when they first saw them, they thought it was the tree of life. Arbor vitae in Latin. They thought every single thing that happens in life, in your personality, happens within the cerebellum and the arbor vitae. They were wrong. All that's happening in the cortex, but at least they named it and it stuck. Gray matter is going to be cell bodies. White matters are going to be axons with myelin. This is going to bring us to embryonic development. Nervous system develops from the ectoderm. If you remember meso, ecto, and endo, ecto is the one on the outside. So your whole nervous system is coming from this outermost layer. Early in the third week of development, the neuroectoderm starts to form, which is a different layer. A dorsal streak appears along the length of the embryo, which means on the back, which just happens to be where your spinal cord is. It's going to thicken, these cells are going to thicken to form a neural plate. These neural plates are going to give rise to most of the neurons and glial cells, except for the microglia because they are coming from monocytes from the blood. And as the thickening progresses, the neural plate sinks, which just means it goes down. So it's starting to form almost a horseshoe. The edges are going to thicken. This will form a little groove. Then the groove is going to have these raised folds on each side, and the folds are going to fuse along the midline. By the fourth week, it's going to fuse together and form a hollow channel. So if you go back to spinal bifida, how does neurons, in spinal bifida involve the fourth week? Because by the fourth week in embryonic development, you don't have like a crest. You now have a hollow tube. So if that hollow tube doesn't become a hollow tube, then the next step of neuronal growth will start to grow outside that tube, and that's spinal bifida. So the neural tube separates from the overlying ectoderm. It's going to go deeper underneath into more tissues, and then you're going to have nerve fibers start to grow. So the neural crest is formed from the ectodermal cells that are going to be on the margin. We're not really going to look at it, but the neural tube is going to have three different parts. Forebrain, the procephalon, midbrain, mesencephalon, hindbrain, rhombencephalon. Make a note card, because these are the beginning of all these awful names and what you need to know. And the reason why, by the fourth week, you start to have a couple different bulges. And then at the end of the bulge, you have a long, tube. The long tube becomes the spinal cord. But the hindbrain, the rhombencephalon, differentiates into other structures. The mesencephalon differentiates into other structures. And proencephalon goes to other structures. And hint, the mesencephalon becomes the mesencephalon, so that one's kind of easy. And I guarantee I just confused you. So the forebrain then divides into two more structures. The telencephalon becomes the cerebral hemispheres. The diencephalon has the optic vessels and becomes the retina of the eye. The mesencephalon becomes a mesencephalon. The hindbrain goes into two more structures, the metencephalon and the myelencephalon. So when I explained this earlier and I said, so do these sound confusing? You're like, yeah. Now when you see it written in red, they are very, very, very confusing. So I think note cards are the best way to kind of handle this. And this is kind of a way to look at the 4th week where you can kind of see this whole thing of what's actually happening, where it's sinking. You can see it's sinking down. That's what I meant by when I was saying it sinks down, and then it has that crest over there of thickened cells. Eventually it sinks all the way down, then it starts to form a hollow tube. So that's why by the 4th week, if that tube doesn't form correctly, all the stuff that happens inside of that tube, if it's further down, it's the spinal cord, it'll start to grow outside of the cells and will become spinal bifida. So here, this is the 4th week, and you can see those 3 different structures, and there's your spinal cord down there. This is kind of what it looks like. Actually, this is what it looks like. It still has a tail, so if you're wondering what that is, that's a tail. You have the proencephalon, the myelencephalon, the rhombencephalide, and then the spinal cord. That's where it's all kind of bent up. At 5 weeks, it's more differentiated. And by the 5th week, you have 5 structures. The telencephalon, cerebral hemispheres. The diencephalon becomes over here. We'll talk about that with the thalamus later. The mesencephalon just stays the mesencephalon. And then the myelencephalon, pons medulla oblongata, is the myelencephalon. So meninges, ventricles, cerebral spinal fluid, and blood supply. The meninges we're going to go over very quickly because we've already talked about them when we did spinal cord. Ventricles are a little confusing, but they're pretty easy once you kind of know how you name them and where their location is. Cerebral spinal fluid is extremely important to the functioning of the brain, so we're going to spend some time with that. And blood supply, we quickly kind of go over that because we look at it in AMP2 when we do blood circulation. So the meninges, again, from superficial to deep, dura mater, arachnoid matter, and pia mater. Dura mater is that tough mother in German, and it provides the structural framework for the arteries and the veins to go around it. Protects the brain and spinal cord. The arachnoid matter and pia mater are similar to those of the spinal cord. The arachnoid matter is just on top of the brain surface. It has a subarachnoid space that separates it from the pia mater, and then that's where you're going to have cerebral spinal fluid. Pia mater, you can't really see it without a microscope, but if you do kind of pick at it with the sharp probe in lab, you will be able to pick some of the pia mater off of the sheep brain. And that's just what it looks like. You have the dura mater, the arachnoid matter, and you have that subarachnoid space. Then you have the dura mater that kind of sits transparently on top of the brain. Meningitis is going to be the inflammation of the meninges. So if you have bacterial or viral meningitis and you're a child, you can die within a few hours. If you have meningitis, either bacterial or viral meningitis as an adult, you can die within about 12 to 16 hours. So you can deal with it a little bit better. What happens, and the reason why children and adults take quicker to kill, is because meningitis, when you're 3 months to 2 years of age, you have less space. There's a lot less room for the bacteria infection to kind of sit. You also don't have all the myelination of all the neurons at that point. So what happens with bacteria or viral meningitis, it goes through your nose, through the mucous membrane, and it goes into and passes into the blood-brain barrier. The problem with that is if it passes into the meninges and your cerebral spinal fluid, you're still going to have an immune response that's going to attack the bacteria and the virus. The problem with that is it's going to have an inflammation. Inflammation in any other part of your tissue, it can grow out. You'll just have swollen joints or swollen tissues. But in the brain, it's held together by your skull and your spinal cord. So if that inflammation keeps pressing on the spinal cord, because it can't go out, it has to go in, it's going to compress the nerves. If it compresses the nerves too much, it stops the nerves from working. And if it stops the nerves from working, you'll pass out, go into a coma. The that you probably can't wake up from. So if to get diagnosed from spinal meningitis, you have to go to the doctor. They have to do a lumbar puncture, which is called the spinal tap. They draw some cerebral spinal fluid out of from the lumbar puncture, and then they test it and they see if there's a bacterial or a viral infection. If you kind of think, doctor's hat again, why would you do a lumbar puncture and not one in the thoracic or the cervical?
Because the spinal cord is not there.
Yep, there's no spinal cord in the lumbar region. So you can quickly, if you're an adult, if you're 3 months old, you have to be very careful when you're doing a spinal tap. But if you're an adult, you would do it in the lumbar region because there's no spinal cord there. When you put the needle in there, the axons would move out of the way and you'll be able to draw fluid from there. These are the ventricles. This is how your cerebral spinal fluid can kind of move around. If you look here, they have these two lateral ventricles, and they look kind of like ram's horns. So you have one on this side, one on this side, so there's a total of two. This next little pool right here is called the third ventricle. Then the third ventricle is connected to the fourth ventricle by the cerebral aqueduct. And then when it goes down to the spinal cord, there's a central canal. There you go. There's your ventricles of the brain. And just if you kind of see it left and right, okay, there's two. That's the one in the middle where you can see the left and the right thalamus and left and right hypothalamus. That's going to be bathed and sitting right next to the third ventricle. So let's kind of look at the ventricles and how you make cerebral spinal fluid. We talked about the two lateral, the third and the fourth, and the cerebral aqueduct, so we can skip that. We're going to go directly to the choroid plexus. Choroid plexus are going to be a spongy mass of blood capillaries on the floor of each ventricle. Spongy mass of blood capillaries. On top of them, they're going to have epidermal cells that we talked about earlier with glial cells. These are going to line the ventricles and cover the choroid plexus. So these epidermal cells, because they cover the choroid plexus, this is going to allow fluid to pass out from these blood capillaries, and that fluid that comes out will become your cerebral spinal fluid. So the choroid plexus are one of the major things that the body has to create cerebral spinal fluid. Yes, there's a lot. It bathes the external surface and internal surface of the brain. Some of it's going to be in the subarachnoid space. Some is going to be in the epidermal lining of the ventricles. Some will be by the choroid plexus. But if you kind of think about it, the general epidermal linings, these are just going to be on the choroid plexus anyway. So the majority of your cerebral spinal fluid will be directly or indirectly made by the choroid plexus. So where do we reabsorb these? So all of this is going to go around and bathe the brain until it goes to the top of the brain, almost in a circular motion. It's going to be reabsorbed by arachnoid villi, cauliflower-shaped extensions of the arachnoid meninges. They protrude through the dura mater and into the superior sagittal sinus, which is the venous drainage of the brain. The cerebral spinal fluid penetrates the wall of the villi and mixes with the blood of the sinus. We'll take a picture of that, and I'll show you why it says it's cauliflower-shaped. What does the cerebral spinal fluid do? Buoyancy. You've all floated in a pool before. You feel a little lighter when you're floating in a pool. If somebody, and when I was doing my master's degree, somebody took a brain out of a Tupperware container, which is just strange to think about it now, and put the brain on a table. When the brain sat down on the table, it kind of squished, and you're like, that's disgusting. And when it sat down, you could see the force of gravity push the brain down. So the cerebral spinal fluid, when it's floating and circulating all around the brain, it's creating a buoyant condition for the brain to kind of float there and not be weighed down by gravity. It also protects the brain. It's going to protect it from going from side to side, up and down, from striking the bones next to it. It's also going to provide a chemical stability. So when it needs ions, there's ions available. When it needs to remove waste, it can remove waste. And here is kind of all of the little things that happen. You can see the choroid plexus with the epididymal cells making it. You can see it being circulated all around and up, back, and around. And you can see these little things up here. Do they kind of look like little extensions of cauliflower? Maybe, maybe not. But that's what those cauliflower little projections into the sinus, and that's how the cerebral spinal fluid goes back into the venous circulation. Veins go back into the heart to be for the waste to be removed and everything to be cleaned and more oxygen to be brought back in. And then finally, the blood-brain barrier. So circulation. The brain is only 2% of your body weight. It receives 15% of the blood. So just because it's so small doesn't mean it's not important. 15% of your blood means that the neurons are going to need a lot of ATP for energy. If it needs a lot of ATP for energy, the mitochondria is going to need a lot of oxygen. And if there's oxygen, they have to create ATP from glucose. So that blood that's being brought to the brain is high in oxygen, high in glucose. Once you learn the circulatory system, the first couple branches off the aorta, directly off the heart, they go directly to the brain. The muscles and everything else was kind of like receiving everything else. So if you have 10 seconds of interruption with blood flow, you will lose consciousness. If you've ever had that crazy person in high school or even college that kind of says, watch me, I'm going to make my face turn red, and they hold their breath for like 10 seconds, sometimes they pass out. The reason why is they interrupted blood flow to the brain for about 10 seconds. If they stay unconscious and they don't have any blood flow to the brain from 1 to 2 minutes, they can start having significant impairment in neural function. If they go for 4 to 5 minutes, then sometimes they won't have any way to go back, and it's irreversible brain damage. So it's very important, even in CPR, to pump the blood to the brain. And if you can keep pumping blood to the brain, oxygen and glucose is going to the brain just enough to keep those neurons from dying. Blood is also a source of antibodies, macrophages, bacterial toxin, and other harmful agents. So there needs to be a way for the brain to kind of limit what actually makes it into the brain or spinal cord. And that's going to be the blood-brain barrier. I mentioned how it does this, but I'm going to kind of go into a little more detail on how it's actually going to do that. So the 2 points, the blood capillaries to the brain tissue, and then the capillaries of the choroid plexus. We kind of talked about the epididymal cells in the choroid plexus and kind of how they kind of limit stuff going in. So we're going to more focus on the blood capillaries going to the tissues. That's called the blood-brain barrier, and it's going to protect the blood capillaries throughout the brain tissues. It's going to have tight junctions between the endothelial cells that form the capillary walls. How do they form those tight junctions? With those astrocytes, those neuroglia we talked about earlier. Those astrocytes have these little feet. These little feet go on the capillary. They start forming these little tight junctions so small things can pass through them. Large things have to stay in the blood. So these induce the endothelial cells to form the tight junctions. So that means blood must pass through the cells and not between them. Keeps harmful substances from passing into brain tissues while allowing smaller and necessary ones to pass by. And finally, the blood-brain barrier, or the cerebral spinal fluid barrier, protects the brain at the choroid plexus, forms tight junctions between the epididymal cells, and those tight junctions are absent from other epididymal cells elsewhere, and they're important to allow exchange between the blood-brain tissue and the cerebral spinal fluid. And the blood-brain barrier is highly permeable to water, glucose, oxygen. But if you think about it, glucose and oxygen are the 2 main ones that you want to keep into the brain because they're ones that they're going to need for ATP. It's going to be slightly permeable to sodium, potassium, chloride, and waste products. They want to keep them out.